The second session is looking about animal equine health and welfare updates, and I'm delighted that Peter Laurie, in his capacity as Non-Executive Director for the Animal Health and Welfare Board for England, is chairing this session. So Peter, over to you.
Thank you very much, Tim, and good morning, everyone. As Tim mentioned, I'm here from the Animal Health and Welfare Board. Some of you will be familiar with its work, I'm sure, but essentially we advise DEFRA and its ministers on strategic issues that affect all kept animals in this country. And really pleased that Jonathan Statham, the board's chair, is with us here today as well. And he and I, and indeed all of our board colleagues, really recognise the significance and also the scale of the equine sectors in this country, as well, of course, as the joy that working with horses, riding horses, and simply being around horses brings to so many people. So it follows to us, to the board, that the health and welfare of our national herd of horses is of critical importance, as it is to the forum here today as well. And it's a real pleasure to chair this next session that's going to bring together three distinguished speakers who are going to provide updates on important health and welfare issues. And as before with the last session, there's a chance for your questions at the end as well. So I do encourage you to feed your questions into Slido, and we'll take as many of those at the end as we can. Our first speaker in the session is Professor Bruce McGorum, who is a professor at the Royal Dick School of Veterinary Studies in Edinburgh. And you can read more about Bruce and indeed all of our speakers in the programme. But Bruce is going to start off by talking to us about equine grass sickness, the latest understanding, including the probable cause, and he's going to outline some directions for future work in this area as well. So welcome, Bruce.
Thank you, Peter. Ladies and gentlemen, it's my great pleasure to be with you today, and in particular to give you some good news about this otherwise devastating disease. And as Peter mentioned, we strongly believe now that the disease is likely caused by a neurotoxic phospholipase A2. And just to unpick this, neurotoxic obviously means that it's injurious to the neurons. A phospholipase A2 describes its action. It breaks down lipids and unfortunately or fats, and unfortunately that involves also the lipids in the key part of the membrane of nerves. And we obtained this evidence when we studied the microscopic detail of the neuromuscular junction. And these cartoons illustrate the neuromuscular junction. This is the microscopic intimate connection between the nerves and the muscle bundle that's responsible for initiation and control of muscle contraction. And when we studied these in grass sickness horses, we identified that these had abnormalities that were identical to the effects produced by this neurotoxic phospholipase A2. Every day is a school day, so this is a little bit of neurophysiology for you this morning. This is how muscle contraction occurs. We have the nerve here and we have the muscle here. And the contraction is induced by the nerve impulse passing down, and when it gets to the terminus of the nerve, it induces these little vesicles, we call these little packets of neurotransmitter, to fuse with the membrane and discharge their neurotransmitters, which pass across this microscopic cleft, stimulating the muscle to contract. Thereafter, these neurotransmitters are reabsorbed back into the vesicles, and these are recycled. The muscle then relaxes. If we look at these neuromuscular junctions under electron microscopy, highly magnified, this is the healthy horse here, this is the nerve in the top part, and this is the muscle, this is the junction here, and we can see the nerve is replete with numerous small, these microscopic vesicles containing the neurotransmitters. In contrast, if you look at a horse with grass sickness, the first thing we see is this ruffling of this membrane. And this indicates the effect of the phospholipase A2 enzyme. This has disrupted the lipids in that membrane, and that firstly results in a massive uncontrolled release of neurotransmitters across to stimulate the muscle, and also prevents reabsorption of the neurotransmitter back into the nerve. And as a consequence, this results in depletion of the vesicles. We can see this is empty of vesicles, and then the nerve ending undergoes degeneration, and obviously this compromises the function of the muscle. We studied one horse with botulinum because there was a lot of evidence suggesting that botulinum was linked with grass sickness, but we found that the changes were the opposite to grass sickness. This does paralyze muscle, but it does by preventing the release of these vesicles. So they end up with a normal number of vesicles in a botulinum horse. So this is further evidence that the disease is not caused by botulinum neurotoxins. And this evidence, for the first time, I think, explains the profound effects that we see in the affected horses. We can see the significant muscle fasciculations in the triceps and in the flanks here. This has been hitherto unexplained. And also this chronic case shows significant muscle weakness. He has a base narrow stance, or an elephant on a tub stance. He's shifting his weight continuously because his muscles are weak. He's resting his hindquarters against the stable to take weight, and he has a low head and neck carriage. And this also a fairly gruesome video, but this is a horse with grass sickness undergoing exploratory surgery just to rule out any surgical injury and also to get a biopsy to confirm the diagnosis. And this shows the problem that grass sickness horses have. There's a failure of peristaltic movement, coordinated movement of fluid from one end of the intestine to the other. It's not a flaccid paralysis. We can see that some areas have, an excessive, continuous, almost spasm, while other ones are fairly relaxed, and this results in a failure of movement of the fluid and digesta through the intestinal tract. And this is explained again by the toxin. So we've identified the effect of this toxin, but where does the toxin come from? The most likely source is a pasture microbe, and we know that at the time of grass sickness, the herbage is covered in a vast array of bacteria, fungi, diatoms, etc., all of which will have phospholipase E2, some potentially neurotoxic. Another possibility is that the plants themselves, they also contain this, so that's a possibility. We know that both the microbes and the plants, if they're exposed to stresses, such as the weather stresses that trigger this disease, they will have higher activity of these enzymes. Another possible source is in the intestinal bacteria and the fungi, and we know that when horses develop grass sickness, they have a significant alteration in the fungi and the bacteria that are present in their intestine. So there are three possible sources. We think that this enzyme also explains the disease which is identical to grass sickness that occurs in other species, most notably hares, dogs, and cats. I termed this lecture Snakes in the Grass, rather enigmatic title, but this is because one of the key toxins in snake venom is a phospholipase A2, and it's responsible for most of the symptoms that snake envenomation is associated with. We don't think there's any association with snakes and grass sickness, obviously, but we can use information from the research into snake venom activity to further understand our equine disease. And if we compare the clinical signs and the consequences of grass sickness versus human envenomation, we can see there are a lot of similarities here. And in particular, this patient here has the characteristic paralysis of the eyelids, something that we also see in grass sickness. Now, there are some subtle differences. The grass sickness horse has a predominant dysfunction of the intestine, whereas the people tend to die with respiratory paralysis, so they have more of a consequence on the muscle. And this probably reflects the different route of exposure in the horse versus the snakebite victim. In a snakebite victim, the venom will be injected directly into the muscle or directly into the blood, where it will target the muscle more clearly, whereas we're proposing that the horses are getting toxin from the intestinal tract and have a more profound consequence in the intestinal tract. And we are collaborating now with workers in what's termed the World Health Organization Global Snakebite Initiative. And this is a group set up to try and address the really unmet needs of people with snake bites across the world. There's a huge problem. I don't know if you can read this data there, but approximately, I think it's 2 million envenomations currently every year. Between 80 and 130,000 people die. So there's a lot of research currently going on. They're recognizing that the current anti-venom treatment has limited efficacy and limited availability, perhaps, and they're trying to develop alternative drug to treat people with snake envenomations, particularly in rural communities. And this is one drug that they're using in a trial this year in Asia, and this has been selected because it has efficacy in a mouse envenomation model. So they inject venom into mice, and 4 hours later they give this drug, and it does improve survival, it improves muscle function, and improves nerve regeneration. And we plan to use this in a small group of horses in a treatment trial later this spring to determine whether this can improve survival of horses, or whether perhaps, unfortunately, the nerve degeneration is too far advanced to make a significant improvement. What else are we doing? We've identified a smoking gun, the effect of this nerve toxin, but it would be nice to actually identify and characterize and determine the source of the toxin. And there's work at Edinburgh and Surrey to try and achieve this. There's also the Morden Research Institute Equine Grass Sickness Fund has set up a biobank archiving samples from horses and pastures affected with this disease, and this is made freely available to researchers in the area. And finally, there's also a very promising predictive weather app by Hayley Coulson at the University of Edinburgh, designed along the similar lines to the Laminitis app to warn owners of high-risk weather that usually trigger off episodes so that they can potentially bring the horses off the pastures to reduce the risk of disease. And finally, I'd just like to thank the significant help that we've had from the equine sector, from the institutes, the funders, the vets, and obviously owners. And thanks for your attention.
Let's move straight on to our second presentation now, and this is from Professor Richard Piercy from the Royal Veterinary College here in London, and Richard's going to talk to us about atypical myopathy. Richard.
Thank you very much. Good morning, everybody. Thank you to the organisers for the invitation to speak. So, Sycamore toxicity or atypical myopathy. The first thing I wanted to get across to you is something that you probably all know. Horses are phenomenal athletes. Let's think a little bit about how athletic expertise can be measured. One of the ways it can be measured is through measuring something called VO2 max, which many people here will be familiar with. What is it? It's the maximum rate of oxygen consumption during exercise. So let's look at some fantastic athletes. Here's Sir Matthew Pinsent. He has when he was training anyway, a VO2 max of 75 mils per kilogram per minute. Here's Jean-Benoit Samuelson, highest recorded VO2 max in a woman, 78.6 mils per kilo per minute. Highest in a man, Oscar Svensson, a Norwegian cyclist, 97.5 mils per kilo per minute. Most people in this audience probably have a VO2 max of around about 40 to 50, okay? Thoroughbred racehorses, 200 to 240 mils per kilo per minute. They are phenomenal at breaking down oxygen with metabolites to produce energy in their muscles. So when things go wrong, the first reports of fatal muscle disease were reported by these authors in the veterinary record back in 1942. And these were horses that developed muscle disease when they were out at pasture, not particularly when they were exercising. And because it was occurring when they were at pasture rather than when they were working, it was known as atypical myopathy. These horses present with severe weakness. They collapse. They struggle to stand up. They have markedly discolored urine. On the right-hand side in that little pot with the green lid, that's urine from a horse because of the breakdown products of its muscles that are passing through its kidneys into the urine. They have severe heart arrhythmias, and the photograph here is a cross-section through the heart of a horse with atypical myopathy. You can see the larger left side of the horse's heart on the right of this image, and it's pale because it's full of fat, and it's pale because it's necrotic. The heart muscle is also affected. These horses have significant difficulty breathing, and in fact, it's either the heart usually or the respiratory muscle compromise that kills these horses. So we now know, of course, through some work that was done by colleagues in Liège, Dominique Vauchen in particular, that atypical myopathy is associated with ingestion of sycamore tree material. Many people here will be very familiar with the sycamore tree. Of course, it has this classic tri-lobed leaf and the very obvious seeds that we know as sort of helicopter-like seeds. Sycamore contains a toxin, which is known as hyperglycin A, and this is the chemical structure of it. So how does this toxin affect the muscle system? Well, in order to produce enough energy for muscle contraction, we need to produce ATP. And ATP in muscle, particularly in horses, predominantly comes from fatty acids derived from the forage in their diet that we've heard about this morning. It turns out that hyperglycin blocks the way the fatty acids are incorporated in the mitochondria, which produce energy, in order to produce ATP. So when hypoglycin is present, we don't produce enough ATP, and that's why the muscles cannot contract and why we have all of these clinical signs. Sycamore is becoming increasingly common across Northern Europe. And this is just some work that was done by a PhD student of mine in collaboration with the Woodland Trust, looking at the numbers of sycamores in the UK in different periods. So on the left-hand side, we've got 1900 to 1950. Actually, very few sycamore trees, relatively speaking. But look what happens between 1950 and 2000, an explosion of sycamore trees across the UK, which has continued in recent decades. Sycamores are like a weed. This is the, I often take the train from King's Cross to Potter's Bar, which is where I work. And this is just looking out of the train carriage window at sycamore seedlings or saplings growing on the sidings. They're everywhere. They're even in the Jockey Club in Newmarket. A few years ago, funded by the Horse Trust, we developed an assay for measuring hyperglycin A in plant material, but also in horse blood. And some of the data that I'm now going to show you is derived from that work done in collaboration with the Horse Trust. In particular, something that's very interesting is if you take the same, in this case, 14 trees spread across the middle and south of England, and measure their hyperglycin A content in their seeds in 3 successive years, you can see that in this case, in 2017, the hyperglycin A content was higher. And one of the active areas of investigation for us at the moment is trying to work out why do sycamore trees produce more hyperglycin A in their material in some years compared with others. We don't know the answer to that at the moment. But of course, that may have a role to play in the susceptibility of horses to the toxin. We also know that hyperglycin A production in sycamore material is a dynamic process. If you take sycamore seeds, grind them up in a pestle and mortar, and then store them in different ways, you can see that the amount of hyperglycin A will go up. And that's what's shown here in this graph on the left-hand side. In particular, if you add some water and store at room temperature, 20 degrees centigrade, so that's the right-hand most bar on this graph, you can see that the amount of hyperglycin A is much higher than if the tissues are stored at lower temperatures. Now, that's in ground-up seeds. What's interesting is that if you just have the whole seeds and store them in the same ways, we don't see the same effect. There's something about the seeds being ground up and then stored with some water and at room temperature or a higher temperature that produces hyperglycin A. It's a dynamic process. And we think potentially this may be a defense mechanism for plants. Potentially hyperglycin A production goes up in the plant material when it's ground up, in other words, when it's being eaten. We know that hyperglycin A remains in sycamore material, in this case seedlings, after it's been mowed for several weeks, as shown here. And in fact, if you cut sycamore seedlings, the amount of hyperglycin A goes up in the first 48 hours, as is illustrated in this graph. We also know that hyperglycin A remains in seedlings if you try to destroy them with herbicides. And that's what's shown here. Now, this is not good news for all the horse owners here who are trying to get rid of these wretched seedlings, okay? But the point is, you do need to avoid the seedlings, and you can't do it simply just by mowing and leaving the seedlings on the pasture or by spraying them. You have to wait for them to completely disappear. The other thing that we've discovered is that if seedlings or seeds are incorporated into hay, the toxin remains in the hay months after the hay is made, or haylage. So what is the risk? Well, of course, the risk depends on lots of different things. We know that the toxin is highest in seedlings compared with seeds, and then the lowest amount of toxin is relatively in the leaves. We know that there is year-to-year variation in toxin production in sycamore trees, and we know that there is significant year-to-year variation in the amount of seed production by sycamore trees, so-called mast years. We've demonstrated that in mast years, in other words, when sycamores across a region produce many more seeds for whatever reason, there's a higher likelihood that horses will get atypical myopathy that year. It makes sense. Horses are more exposed to the toxin productive material. We know that toxin concentration varies in the sycamore material year on year. Of course, it may also depend partly on your horse's grazing habits. I think it's possible, although I don't have any proof for this, that some horses actually like the taste of hypoglycin A. Maybe they actively try and eat this stuff. And of course, a lot depends on just how much they eat. What about prevention? Well, obviously, try to avoid sycamore trees, especially in late autumn when there are seeds on the fields, and in the spring when the seedlings are out there. Feed from hay racks. It makes sense. If you're feeding from a hay rack, the horse is not going to be potentially eating seeds that have got onto the ground. Bring in horses at night. Potentially, if horses are grazing overnight, they might be eating the seeds and not knowing about it. Quite a lot of people are cutting down sycamore trees. I get a lot of telephone calls about disputes between yards and field owners and horse owners, people wanting to cut down sycamore trees. Now, I'm not going to legislate in this area, but it's a big, big problem, and some people here may have come across this. It won't have escaped your notice that there is an irony here, of course. Quite rightly, there was national consternation about the sycamore tree gap, but there are other people cutting sycamore trees down. And the sycamore is actually not a native tree to the UK. It was introduced to the UK in the 1500s. So I will finish there. I want to acknowledge again the Horse Trust, who've supported all of our work, and the London Bioanalysis Centre, who we work closely with for the analysis itself. Thank you very much.
We now come to our final speaker in the session, Dr. Julia Shrubb. And Julia is both an equine vet, but also the chair of CANTER. And as we were reminded in the quiz at the start of the day, CANTER was launched here at the Forum back in 2023 to help combat antiparasitic resistance in equines. And 2 years on, we're delighted to hear from Julia, who's going to give us an update on CANTER's recent work. Welcome, Julia.
Brilliant. Thank you very much. Thank you, everybody. I'm delighted to give a brief update today. So those of you who aren't aware of CANTER, and I know many of you will be aware of it, in fact, we've got many members in the room here today. We are a diverse group of people from all different sectors of the different parts across the equine industry that have come together to try and tackle the very real problem of anthelmintic or dewormer resistance in our horses. So I'm excited today that we've got 2 key updates to give you. But first, I thought I'd have a look back at the CANTER journey so far and put it into the context of this forum for those of you who are regular attendees. So if you want to cast your mind back, I haven't got a picture on here, but back to 2020, Dave Rendell gave an excellent presentation outlining the problems of anthelmintic resistance. His talk was entitled An Issue of People or Parasites. And he outlined that if we continue doing what we are doing, we are heading for a welfare crisis where no wormers will work. So there was a lot of discussion about, for many reasons, that we have very much a fragmented industry. And despite the fact that experts have been advocating diagnostic-led worming and not just blanket worming for about 30 years now, we, the equine industry, are not doing a very good job of it. So he and others at that time appealed to the industry for help, which is exactly what happened. So in 2021, initiated by the Veterinary Medicines Directorate, there was a stakeholder workshop in November of that year. The VMD invited people from all across the equine industry, brought us all up to speed on the state of play in the UK and abroad, and discussed what barriers and potential solutions we could come up with. Then in 2022, Alison Pyer and Claire Stratford, from the Veterinary Medicines Directorate gave an update here with the key outputs from that workshop and outlined plans for what was to become CANTER. That was part of an excellent theme session, if you were here, entitled Equine Anthelmintic Resistance, A Collective Responsibility. We heard from Dave Rendell again. We heard from Claire Shand, who leads the marketing for CANTER and is in the audience today, as her perspective as a RAMA. And there was a lot of discussion. One of the key points from the discussion is there's lots of inconsistent and confusing messaging out there. We need an approach that is now much more nuanced. There is no one-size-fits-all when it comes to responsible dewormer use. So there was a call for greater dialogue between horse owners and prescribers. One of the other key take-homes from me in that session was Dave Rendell mentioned that education alone doesn't work. We preach to the converted, as I fear we'd be doing today if we were giving you a talk on exactly what to do. So then, during 2022, CANTER began to take shape. So the first core steering group met and other subgroups were formed. And then, as we all now know, and we're not going to forget that here in 2023, 2 years ago today, Claire Stratford stood up here and officially launched CANTER. Put this motif up just to remind everybody that CANTER has representatives from all across the equine industry. It's not just prescribers, so RAMAs, SQPs, pharmacists, vets. We have parasitologists, experts in their fields. We've got diagnostic providers, representatives from trade, representatives from the pharmaceutical industry, charities, horse owners, competition bodies, regulators, and we even have behaviour change scientists involved. So when CANTER was launched, these were the key priority activities that were outlined, and I'm delighted to say that all 4 of these have been realised. And my first key update for you today is this last one, that in October of last year, Alison Pyatt formally launched these CANTER guidelines on the international stage in France at the International Equine Infectious Disease Conference. That was followed by a very lively and interesting discussion and stimulated a lot of discussion among the industry. So these guidelines have been a lot of work. They're split into handy chapters written by experts in their fields, but they're aimed at prescribers. But they are freely available for all on the internet. The idea being that we can come up with a consistent approach, a little bit of consensus across the industry, and provide practical evidence-based principles to try and slow wormer resistance down. There's also a chapter in there on the environmental impacts of these drugs, as well as an entire chapter dedicated to behaviour change. So there are lots of key things that I could pull from these guidelines, but today I just wanted to highlight this risk profile table, which many of you will have seen before. This really helps with that nuanced approach and helps with that dialogue between prescriber and horse owner. So experts are in agreement that no dewormer should ever be given without risk assessment and/or testing. So I'm not going to go through all this now, but if you have a young horse under 5, it is automatically in that high-risk category. It might be that larvicidal moxidectin treatment over the autumn-winter is absolutely justified without any specific testing. On the flip side, if you have a horse that fits all the low risk in the low risk column and is very well managed and is regularly tested, you probably can't justify giving any wormer at all. So I'm going to ask you to do 2 things, if I may, today, is after this, perhaps on your way home, perhaps over a cup of coffee this weekend, have a look at this table. It's freely available on canterforhorses.org.uk. And the second thing I would really like you to do is to tell somebody else about it. Because as I say, we preach to the converted, but if you can share this or talk to somebody that is not so engaged in this subject, many of us will be very grateful. So what's next? So getting those guidelines out for prescribers was a monumental effort, and we're delighted that we can do that. But the next phase is getting the message out a bit more and really looking at our communications. And we'll be really concentrating on getting the message out to horse owners. Just to say, those guidelines are a live document and will be updated as and when necessary. And there's a couple of key points that we will improve as well. But my other key update for you today is formalising our plans for governance moving forward. So you will remember the Veterinary Medicines Directorate were the initiators of this. They have been an absolute driving force. And without them, CANTER would not be where it is today. They are unable, though, to offer this level of support long term. So today at the National Equine Forum 2025, I'm delighted to announce that we will be moving forwards, working with partnership and under the umbrella of RUMA, Companion Animal and Equine. So many of you will be aware of RUMA, but it stands for the Responsible Use of Medicines Alliance. And to date, they've mainly concentrated on antibiotics, but anthelmintics is clearly the next step. CANTER will keep its own identity, but RUMA, their values and approach align with ours, and they are able to offer us administrative support, financial management, and excellent governance. So, with my quick update, I'd like to finish, and I don't quite know how much time I've got, by just acknowledging the hard work and dedication of everybody within CANTER. There are lots of people in the room today, as I said, but in particular, I would like to acknowledge the three members sitting in front of me from the Veterinary Medicines Directorate. We have Alison Pyer and Claire Stratford, previous chairs of CANTER, and Jodie Perret, the outgoing Secretariat. Without their hard work and dedication, it would not be where it is today. And hopefully you've been a large part in slowing wormer resistance and hopefully improving the welfare of horses for generations to come. So if you could all just join me in showing our appreciation. Thank you.
I'd like to start, as in the previous session, with a question hopefully from the live audience in front of me. Has anyone got a question they'd like to put to the panel? Please do wait for the microphone, sir, but if you could state your name, that would be very helpful. Thank you.
Yeah, Mark Weston from the British Horse Society. The government is funding a dramatic programme of tree planting across the country at the moment, and I wonder whether the industry is or should be making representations to DEFRA as to the appropriateness of the planting of sycamore trees within those planting programmes near to horse pasture or pasture that's being used to produce hay.
Mark, thank you very much for your question. Really important observation. I'll let Richard go first on that one.
Well, thanks for that question, and a very interesting question. I think at least the people who are involved with these sorts of initiatives should be aware of this. It's quite interesting that I think many people still are not familiar with the possible risk of sycamores. And of course, the sycamore tree is a beautiful tree. There's no doubt about it. And in the right place, they are fantastic, and they provide wonderful landscapes and so on. I think for horses in particular, they are clearly a big problem. We're spending a lot of time now working on other associated risks with atypical myopathy. It's not simply eating sycamore material, it can be other things too. But yes, I think the people who are involved with these initiatives should at least be aware. And yes, we want sycamore trees in our landscape, but possibly not near our horses.
Thank you, and I think wearing my DEFRA hat, Richard, I'll take that one away as well, and perhaps we can work together to try and join the dots up within DEFRA on this important issue. So thank you for that. Let's go to Slido now, and a question for Bruce, or at least for you to start off, hopefully, Bruce. Could climate change be affecting fields and growth, and could that be a factor for grass sickness?
I think there's a strong weather influence. I don't know how climate change impacts on that, but the work I mentioned looking at the weather app has shown that it occurs largely when anticyclones come across the country, and they tend to come across hitting the east coast, northeast coast particularly, and that induces a period of dry weather with a lot of day to night temperature changes, and that seems to be the trigger for this disease, presumably acting as a stress for the grass and or microbes that are growing on that. So if climate change enhances the anticyclones that are coming across, and I don't know enough about that to comment on that. That certainly might impact on the incidence of the disease.
Thank you. And just stay with you, please. How prevalent are cases of grass sickness?
Well, up in the, it's thought to affect about between 1% and 2% of horses in the country, and there are obviously some hot spots in the country. There are higher incidences in Scotland, but there are actually a larger number of horses getting the disease in England because of the larger horse population in England. So it's a significant problem, and it is a recurrent annual problem for some premises, and you can imagine how significant that is in not only in horse welfare, but in the welfare of the people that are trying to make a living from horses and rear young horses. It's a devastating disease.
Right. Thank you. Any other questions from the audience here? Yes, down the front here. Thank you. Microphone on its way, I think.
I've got multiple. It's Siobhan Dillon from Applied Equine Podiatry. I have multiple questions for everyone, but I'll choose one, and it's on the worming side of things. One of the questions I've had in my mind for a long time is there any way of determining worm burden in the field by doing some sort of measurement of worm larvae actually in the field? So if you're doing detection.
I'm not sure I'm the best person to ask this, because I think there is I've certainly read papers from a long time ago, but I'm not aware of any commercial way of doing that at the moment. If anybody wants to answer differently, I will open it to the floor.
Anyone want to add to that this time? Good question, thank you. Any more from the audience before we go back to Sara? Yes, right at the very back, thank you.
Hello, my name is Claire McCafferty from Premier Nutrition. I was just wondering if there was a trend in the incidence of grass sickness, and if that's changed over time. Are we seeing more cases now or less, or is it sort of stagnant since we first knew about the disease?
Yeah, that's a really interesting question, thank you. So just if anybody didn't hear that, it says, has there been a kind of temporal change over the years in the incidence of grass sickness? Well, the disease was first reported in 1906, 1907, and a fairly small number of cases there. They thought probably there had been a few cases prior to that, but it was only in 1906 that it was actually recognised and named. And in those subsequent years, it spread very quickly from the east coast of Scotland right across Britain. And in the 1920s, 1930s, it was a major problem. They had incidences up to 20% of horses on certain premises, and that had a devastating effect on agricultural horses, which were the beasts of burden at that time, and it's said that hastened the introduction of the tractor into North Scotland earlier than elsewhere. So the incidence has been much higher. Previously we're talking about an incidence probably now about 1%, 2%, but perhaps a lot less than the 20% that was reported. And this seems to be what happens whenever it goes to a new country. So whenever a new country reports it for the first time, we get a much higher incidence, and then the incidence seems to diminish thereafter, presumably because the horse population is developing some immunity against the disease, and kind of things are stabilizing there, because we know that immunity is a key thing in this disease, and that if you're growing up in a premises that has had the disease before, there's some protection. If you don't succumb to the disease, you develop some protection. So that's presumably been explaining why the incidence has gone from 20% down to a fairly low level now. And as Richard mentioned with atypical myopathy, we have some years that are a lot worse than others, again, probably reflecting differences in weather patterns affecting grass growth, etc.
Bruce, thank you. I'm going to try and squeeze in two quick questions if we can. The first one for you, Julia. A delegate's commented that FEC and tapeworm saliva tests are becoming more expensive. In that context, how can we encourage horse owners, horse carers to test before worming when it can often be cheaper to just blindly treat their own animals?
That's often what you think when you're faced with the horse in front of you in that test. But overall, doing this approach saves you money. So if you look at any yard, overall they will spend less money. But for that individual horse owner, they might have to test and worm. But overall, they will absolutely spend less. And isn't everything getting more expensive?
Important take-home message, and do test before you administer. And our final question, which I'm going to put to all three, since we've got three distinguished vets alongside me. This is a question from Charlotte. Are any of you worried, or should we be worried, about avian influenza getting into the equine population? And I guess that question follows some reports from the States in particular, where AI has been picked up in other species. Any thoughts or concerns from any of my veterinary colleagues here?
I'm going to answer first because of the Donning-Kruger effect. I don't think I have enough knowledge, so I'm not worried.
Good answer.
I think we should put that to Richard Newton, who's in the audience. But my understanding is that equids don't get the avian influenza, is my understanding, yet. Is that right, Richard?
Well, I'd also defer to Richard Newton, who's in the audience.
There he is. No pressure, Richard.
Thank you. Richard Newton here from Equine Infectious Disease Surveillance. This is very topical. There has to be a concern. The flu that we've been dealing with since 1963 came from birds. This H5N1 that's in this country at the moment and across the world has shown great propensity to get into other animals we wouldn't expect it to. Last year it was discovered in dairy cattle. We would never have thought that. It's made flu virologists think again. So there has to be a concern, and it is something we're monitoring. There was some very limited data came out of Mongolia, actually, where a few horses had supposedly evidence by looking for antibodies against the virus, and that was there. So it's something we need to keep an eye on. I think the risk is very small, but we as humans are also at risk. And flu in birds transferring into other species is a concern, and we've got to monitor it. And that's why we need to be able to, should the worst happen, we need to then be able to be in a good position to deal with that, and I'll touch on that in my presentation. Thank you.
Great. Thank you, Richard. So we're on our guard there. Well, look, ladies and gentlemen, we're out of time, sadly. I know we've got lots of other questions we could have taken, but thank you for those. I'm going to have to bring the session to a close with a huge thank you to our three speakers, to Bruce, Richard and Julia. Thank you for brilliant presentations. Thank you for your engagement and questions too, and I'm delighted to hand back now to Tim. Thank you.
Thanks very much, Peter. Fascinating session. I've learnt a lot about all sorts of things. I had no idea the spread of sycamores over the period. That was amazing. And I'd like to reassure people that, speaking as the treasurer of rumour, we've A, got good governance, and B, we got plenty of cash as well. So it's in good hands, I assure you.